Hello, everyone. I'm Mersa Mardikorayan from Boarding Lab, and I want to talk about data-driven computational protein design. So in this aspect, we want to generate proteins that are not found in nature, but they have our desired function. So a good asset of this paper is that it has a great categorization of different type of data used in machine learning and deep learning algorithms in protein engineering. The first one is data from multiple sequence alignment. And they talked about covariation and that how we can use quantitative analysis of covariations of residues in order to produce distant maps from residues. And lots of pioneers in deep learning like AlphaFold are using that. Another one is using multiple sequence alignment with structure-based modeling in order to identify disruptive residues, and as a result, focus the design on functional mutations. The last one just uses the multiple sequence alignment, and it's called consensus design. It selects the most frequent residue at each position and produces the protein sequence. And surprisingly, most of the studies shown that some candidates that are produced with consensus design have more stability than natural variants reported. Second type of data is data from structure. Design-based, like Rosetta Foldex, use scoring functions to assess the contributions of amino acids in protein stability. Another one is called tertiary structural motifs or terms, that it finds recurring motifs no matter what the protein family is. So in overall, it takes protein backbone coordinates as input and uses term-based sequence structure statistics in order to analyze that configuration. And the last one in structured data is graph neural network or 3D convolutional neural network that learns the residue-residue interaction. The third part is data from experiment. Why we need data from experiment is because lots of structure-based methods had really great success, yet they didn't have good success on difficult problems. Also, multiple sequence alignment is not always available in lots of protein sequences. So we are feeding the algorithm with the data that we are producing in order to guide rather than identify in most cases. So we fine-tune or define scoring functions, or we can guide the library space for more effective mutations using experimental data. So in the horizon, they talked about how NLP got the trend and using embedding methods, compressed vectors showing the representations of amino acids, no matter what the target is, in order to increase the generalizability. This paper has a lot of great references that I encourage you to take a look at, and thank you for listening to me.